how do we study this in the lab? How do we study lab? It's a lab-based. What I've done is, a lab-based kind of cancer research. So what do we do in the lab? So there's two types of samples we can use. So in vitro, so cancer cell lines, and in vivo. So in vitro in Latin essentially means in glass, I think. Correct me if I'm wrong. So this is the most common. So this is these are cells taken from cancer patients historically, and then they've kind of modified it so they become immortal. So they will divide indefinitely, basically. So you can use them in the lab. Most labs in the world will have them. So they can be cultured indefinitely and used in cancer labs globally. And if you go into cancer research, one of the most famous cancer cell is called the HeLa cell. And so this is actually from a lady called Henrietta Lacks. So this is a really good book if you want to read about cancer cell lines. So she was, I think, born in 1920 and she died in 1950 of cervical cancer. And they've taken her cells, modified it, and actually it's now one of the most common cancer cell line used in the labs globally. And then the next model that we can use to study cancer is in vivo animal models. So mice, rats, rabbits, I think they can do dogs as well, but these are the things that at least I have worked with mice. So this mice is different because ito cells lang to, but this is an entire living organism, so a different environment. And so this is better because, as you can imagine, in a dish, it's only cells. It's not really representative of how a tumor is inside a human body. So this is kind of the next level up. Animal models. Ang disadvantage nito is it's very expensive. I think if I remember when I was doing mice work, it was 50 pounds per mice, which I don't know how much that is in pesos. Maybe it's like 2,000. I don't know. But it's very expensive, and it's slow throughput. But again, the answers, the question and the answers that you get from using animal models are very different to what you can get from just using cells in the lab. So this is a cell and this is a cute little mice, but there's different types of mice as well. But yeah, cool. And then what do you need? So the basics of a lab for cancer research. What tools do you need? As I said earlier, so firstly, you need the study sample, the cells that you work with, or the animal models. But how do you actually start studying? How do you kind of culture these cells in the lab? So firstly, you need tissue culture plates and flasks here. So to grow the cells. And then you need some culture media. So this is basically their nutrients. It's a soup. It contains nutrients, proteins, amino acids, and everything that you need for the cells to keep growing. So we use different types of culture media for different types of tumor cells. Some of them are, kind of okay lang kung glucose or something, and then some of them are a bit more high maintenance is what we call. You have to have more growth factors in them, things like that. So you have to optimize the soup to enable these cells to grow happily. Then you need also a humidified chambers, because you can't just leave the cells on a desk. They will just die because it's going to be cold and things like that. So you need to, this is a humidified chamber. So this will maintain an atmosphere for the cells, a constant atmosphere, 5% CO2 and 37 degrees Celsius. And then finally, you need kind of how to work with these cells to keep them sterile. You need some kind of laminar flow hood. So you see these people, I'm sure you've seen this in movies and things like that, the sci-fi movies, when people work with these gloves and things like that. So this enables you to work with these cells in a sterile environment. So to avoid any contamination with bacteria, fungus, yeast, and things like that. So these are just the basics. I mean, there's a lot more tools that you can use, but I just want to give you a flavor of what we use in the lab.